হ্যালো সুকেন্দু।
হ্যাঁ স্যার।
হ্যালো।
হ্যাঁ স্যার। স্যার তাহলে আমরা
হ্যাঁ,
কি শুরু করবো স্যার?
না 10:30টায় টাইম তো।
হ্যাঁ স্যার, হ্যাঁ স্যার,
সাড়ে
অভিজিৎ বাবু
10:30।
জয়েন করে গেছেন।
কোথায় অভিজিৎ দা, কোথায়?
হ্যাঁ
এ
স্যার।
অভিজিৎ,
হ্যাঁ, জয়েন করে গেছি।
ভালো আছেন তো?
হ্যাঁ, ভালো আছি, ভালো আছি।
আপনাদের আপনাদের এই এলাকায় আমাকে ফেলে দিয়ে তো বিপদে ফেলেছে সুকেন্দুরা।
কেন, আপনি কি নিউট্রিশনের লোক নন?
নিউট্রিশনের লোক তো সবাই,
তাহলে
হ্যাঁ, কেউ বাদ নয়, কিন্তু এখানে কথা বলা আপনাদের মাঝে খুব বিপদ।
আরে আপনি তো আমাকে বিপদে ফেলে দিয়েছেন। আমিও তো নিউট্রিশনের লোক নই।
না, আপনাদের সঙ্গে ক্লোজ রিলেশন আছে, আমাদের সঙ্গে মিনি।
রিলেশন রিলেশন আছে, কিন্তু রিলেশনের এখন যা গভীরতা এবং ব্যাপ্তি হয়ে গেছে।
হ্যাঁ, খুব ব্যাপার। দেখি আমি একটু যা পারলাম সাজিয়ে দিলাম, আর কি করবো।
আচ্ছা, স্বপনবাবু, আমার একটা ওই 11:30 থেকে একটা মিটিং শিডিউল হয়ে
আমি
আছে।
10:30 ঢুকে যাবো, 10:30 স্টার্ট করতে বলবো
আচ্ছা,
আপনাকে।
আচ্ছা।
11:10 মধ্যে ছেড়ে দিতে
হ্যাঁ,
পারবো
না,
আমি।
আমি যেটা বলছি, আমি আমি বেরিয়ে যাব, সেইটা আপনার কাছে একটু অনুমতি নিয়ে রাখছি।
না না না, অবশ্যই অবশ্যই অবশ্যই, আপনি যে এসছেন আমাদের এই, এরা এরা আপনাদের ছাত্ররা অনেককেই জোগাড় করেছে, হ্যাঁ, অনেকেই রাজি হয়েছেন, এটাই খুব আনন্দের বিষয়।
হ্যাঁ, হয়তো আমি সন্তোষকে দেখতে পাচ্ছি, ও তো পুনে আইদারে।
হ্যাঁ, সন্তোষ আছে, আপনাদের সুমেধাও আছে।
হ্যাঁ, সুমেধা আছে, সব।
তারপরে আপনাদেরই ছাত্র নাকি, ওই যে আমাদের এতে আছে, ইউএসএ তে, মৌমিতা।
হ্যাঁ, মৌমিতাও আমাদের, সায়ন্তী আমাদের অ্যালুমনাই।
স্যার প্রেম আছে, মৌটুসি আছে, সবাই আছে।
হ্যাঁ, এদের সামনে বলা আর কি খুব চাপের ব্যাপার, বুঝলেন?
তাহলে আপনার যদি চাপ হয়, আমার অবস্থা ভাবুন।
না, না, আপনি তো
হ্যাঁ, আমি তো উপরে উপরে ভাসিয়ে দেবো।
না, ভালো, এটা খুব ভালো ভালো উদ্যোগ নিয়েছেন, মানে খুবই ভালো। মানে এরকম
টাইটেলটা আমরা তো, ওই ফুড হ্যাজ এ মিনিং, দো ফুড হ্যাজ মেনি মিনিংস, কিন্তু
হ্যাঁ,
আমি ফুড হ্যাজ মিনিং বলেই করেছি। মানে সুকেন্দুদের বললাম যে এটা একটা খুব সহজ টাইটেল করো, হ্যাঁ, দিয়ে ওরাও এগ্রি করলো।
না, ভালো। এতে মানে এই বাইরের থেকে এরা আছে, আমাদের মৌমিতা, তারপরে এবং আমাদের এই সন্তোষ আছে, সায়ন্তী আছে। এই ছাত্রছাত্রীদের খুব উপকার হয়, মানে তারা জানতে পারে যে অন্যান্য ইনস্টিটিউট অন্য জায়গায় কিরকম কাজকর্ম।
শুনুন, আপনাদেরকে দেখতে পাওয়াই একটা ভাগ্যের ব্যাপার ছাত্রদের কাছে।
না,
আমি
না,
বলেছি,
এটা বলবেন না।
স্টুডেন্টদের কাছে, হ্যাঁ, স্টুডেন্টদের কাছে তো দেখতে পাওয়াটাই ব্যাপার না।
আর এখন এই সব অনেক কিছু জানতে পারছে, এবং তাদের সঙ্গে কানেক্ট রাখলে ভবিষ্যতে তাদের অনেক কিছু কাজে লাগবে।
স্যার আমরা কি শুরু করবো স্যার? 10:30 বাজে।
হ্যাঁ, করো, করো, করো।
Good morning to all. Hope you all are well in this pandemic situation. It is our great privilege to announce that we are here to participate in International Lecture Series on Nutrition organized by Internal Quality Assurance Cell and Department of Nutrition, Bhushpara Mahavidyalaya. We started the program as quotes of one child, one teacher, one book, one pen can change the world. We are very glad to have some dignitaries who are not only great teachers but also great human beings and great motivators. I am very happy to announce that today we have Professor Abhijit Majumdar, our honorable guest, Professor, Department of Zoology, and ex-registrar of the University of Bardwan. Mr. Sabyasachi Chakraborty, diplomat of American Board of Toxicology, senior consultant, Toxmine BBBA, Brussels, Belgium, and Dr. Sumedha Roy, research scientist, Department of Biomolecular Medicine, Ghent University, Belgium, and she was former assistant professor of geology, the University of Bardwan with us. Now first, I would like to invite our honorable principal, sir, who is the backbone of this event, for inaugurating the program with his valuable words. Over to you, sir.
Thank you, Sukhendu, for your easy introduction. Respected Professor Abhijit Majumdar, Dr. Sumedha Rai, Sri Sabyasachi Chakraborty, and other resource persons from our country and abroad. Friends, it is a tough time to all of us, especially very tough to learners, teachers, and researchers to engage themselves with learning, teaching, and research activities in its proper tune. In spite of all those, it is a pleasure for Guskora Mahavidyalaya and to me to inaugurate this 14 days online international lecture series on nutrition with a very simple but significant title, Food Has a Meaning, organized by the IPSC and Nutrition Department, Guskora Mahavidyalaya. We have with us Professor Dr. Abhijit Majumdar, Professor, University of Bardwan, and an eminent scholar of the subject. We have with us Dr. Sumedha Rai, earlierly assistant professor and presently a brilliant researcher of Ghent University, Belgium. We have with us Sri Sabyasachi Chakraborty, a brilliant consultant in this domain, DAVT, and others from different universities, Professor Dr. Santo Datta, Professor, Department of Home Science, Calcutta University, Dr. Kazi Laila Khaled, Associate Professor, Department of Home Science, Calcutta University, Dr. Moumita Datta, Research Scientist, University of Washington, USA, Dr. Anulakho Chatterjee, Assistant Professor, CU, Dr. Prem Rajak, Assistant Professor, KNU, Ms. Moumita Mandi, Assistant Professor, BU, Dr. Sayanti Poddar, Assistant Professor, MCASC Pune, and Dr. Santosh Poddar, Senior Technical Officer from ISHAR Pune, and many more in this seminar. I most heartily welcome Professor Majumdar, Dr. Sumedha Rai, Sri Sabyasachi Chakraborty, and all others, along with my colleagues of Guskora Mahavidyalaya, my beloved students from this college and others college. The opening day of this series, 1st July, holds a special significance. It is National Doctors' Day today. On this very special day, we are expressing our indebtedness to the doctors community for their dedicated service to the nation with the opening of an online international lecture series on nutrition. Envision food has a meaning. Nutrition is not my subject, and I have very, very limited knowledge in the subject. To me, nutrition is a multidisciplinary subject. Its close relation with physiology, biochemistry, zoology, botany, agriculture science, food production, procurement, processing, medical science, even commerce, economics, and statistics. Like any other subject of study, it is specially designed to develop subject-specific skill in different areas and perspectives. In 400 BC, the Greek physician Hippocrates, the father of medicine, said, "Let thy food be thy medicine, and thy medicine be thy food." Hippocrates realized impacts of food on a person's health, body, and mind to help prevent illness as well as maintain wellness. In this particular moment, I must pay my respect to Anthony Lavoisier, father of nutrition and chemistry. As we become increasingly aware and concerned about nutrition's impact on health, the fields of nutrition science have seen unprecedented growth and expansion in both academic field and practical life. I hope not any academic subject has so close relation as between human life and nutrition. Subject nutrition as a subject of study is not so old as its practice. In common man's life, nutritionists and dietitians, still our grandmothers, mothers, and aunts, they followed the very basic norms of nutrition. The very basic norms of nutrition. But the dietitian and nutritionist entered into the healthcare system and hospital as early as 19th century. Now the knowledge and skill of nutrition has become a profession, and I hope the scope of subject will increase day by day. Dear students, utopia is a sweet dream, but make it happening. First, try to take it as a vision. Vision is nothing but illumination of solitude. Once you have vision, you must try to transform that vision into reality by efforts, by application of minds, by dedicated work, and as a devotee in acquiring knowledge and practicing it in reality. The students, dear students, and I believe the students and teachers of the nutrition department, as well as me also, it is lucky enough to see Professor like Abhijit Majumdar, Dr. Sumedha Rai, Sabyasachi Chakraborty, Shanto Datta, and others, and also to get the opportunity of learning, hearing, and learning from them. It is amazing. Finally, I hope this online international lecture series will be enchanting to the learner, researchers, and others. I do expect a grand success of this international series. Thanks to everybody. Thanks to all.
Thank you, sir, for your valuable word. Now I am requesting Sri Taroknath Datta, Coordinator of Internal Quality Assurance Cell, Mushkara Mahavidyalaya, to introduce our honorable guest, Professor Abhijit Majumdar. Over to you, sir.
Good morning to you all. I feel privileged to introduce our keynote speaker, Dr. Abhijit Majumdar, Professor, Department of Geology, the University of Burdwan, and also former Registrar of University of Burdwan. As an eminent senior academician, Professor Abhijit Majumdar held numerous academic and administrative positions during his long career. He was a visiting scientist to University of Florida, USA. He was also a former research associate, CSIR. His area of specialization, among others, includes entomology, systematics, and biology of Diptera, etc. Professor Majumdar contributed a lot in an international training program on blue tongue vector identification. Professor Majumdar has contributed a number of research articles both in national and international journals. Many students got their PhD degree under Dr. Mojumdar's guidance. He is also a reviewer of a number of international journals. Professor Mojumdar successfully implemented a number of research projects. I hope Dr. Mojumdar's keynote address will immensely benefit both our students and teachers. Thank you, sir. Dr. Abhijit Mojumdar, sir, please.
Very good morning to all. I am really delighted and honored to be invited as a keynote speaker in this inaugural program by Guskara Mahavidyalaya. And I am really very thankful to Professor Swapan Kumar Pan, principal of this college and a patron of this international lecture series on nutrition, which is being organized by IQAC and Department of Nutrition from 1st to 14th July. Professor Pan has been an administrator par excellence, and during my short tenure as a registrar of Bardhaman University, I had the opportunity of having him in several important committees, and his advices and suggestions that I will not forget. And I am thankful to you. And I am very thankful to Professor Dotto, coordinator IQAC, for introducing me with those kind words. Dr. Saurabh Sarkar, convener, he has been right from the day one, time to time updating me about this program. He's in charge of Department of Nutrition, and he has been coordinating. And I thank all the committee members, organizing committee members. And beside, I find there are a lot of participants, student participants, number of research scholars and faculty members during this program. Actually, what I feel that I consider myself a bit fish out of water, I can say. And when I found my name in midst of the distinguished and erudite speakers like Professor Dotto from Department of Home Science, Calcutta University, Dr. Khaled, Calcutta University, Dr. Chatterjee, and besides, I am delighted to find my old friends, Mr. Sabyasachi Chakraborty, Senior Consultant ToxMind, Dr. Sumedha Roy from University of Ghent, an alumni of our department, and he was my next door colleague here, and we have shared some very important research and others with her in a technical discussion. And I find Dr. Moumita Dotto, research scholar, University of Washington, and also an alumni, Dr. Prem Rajak, assistant professor, Kazi Nazrul University, also alumni, Ms. Moutusi Mandi, assistant professor, Bardhaman University, Dr. Sayanti Poddar, alumni, now as assistant professor in MCASC, Pune, and Dr. Santosh Poddar, senior technical officer in a very prestigious institution of IISER, Pune. And with this list, I am really, bit worried and what should I speak and what not. I'll begin with a very small story that once one columnist, a very famous columnist, he wanted to make an article that, most of the brilliant minds, scientific minds, belong to a particular area in southern states. And he was trying to find out, about Professor C.V. Raman, Professor Chandrasekhar, and all, like that. So he went to Madurai to find out, and from Madurai he went to the villages where they lived, and he was talking with their relatives, how was C.V. Raman and Chandrasekhar, and those persons like that. And then ultimately he went to the schools that Professor Raman and all these luminaries went. There, a particular teacher told him, you are a columnist, you should visit their house, ancestral house, and ask their grandmother or any elderly people there that what Raman and all these persons and luminaries used to eat. What was the diet? Surprisingly, the columnist wrote that one common vegetable that they were taking was vendakai. Now, vendakai means okra, বা ভিন্ডি, বাংলায় আমরা যেটা ভিন্ডি বলি. So the columnist, penned down that is it okra the brain food? So please take it as a story, a narrative. So that is the topic that has been chosen. So I'll put it, one change is the diet. So it is a small story. So a diet can change the persons at an individual level, at a species level, at a populational level. And I found that objectives to be quite apt. And what I really miss in most of the biological disciplines, whether it is nutrition or medical sciences or physiology, biochemistry, if you look at biology, one that is lacking is our understanding of evolutionary biology and linkage with that subject. And particularly, it is the theory of natural selection. Because as a student of biology, all of us will agree that it is shaping the functional design of the organisms. So if you look into the concept of adaptations, which allow us to combat pathogens, like we are now combating pathogens, adaptation of pathogens that counter our adaptation. So that has to be understood. And then, what is the mismatch between the body's design and our environment? Are we taking a lot of toxin with our food? And what these toxins are doing into our system? The problem, the crux of the problem is that we are also biological objects and we are also evolving. So but all of us will agree that this evolutionary process is the longest and the most complicated algorithm ever written. It has to be deciphered, it has to be understood, but not neglected by the biological disciplines. Surprisingly, in most of the basic and applied disciplines that has sprouted in the last decade, where interdisciplinary marriages has taken place. And unfortunately, it completely neglects this area of biological discipline, that is the evolutionary biology. Like I can give you an example, if you look into any of the medical sciences curricula throughout the world, nowhere they will teach evolutionary biology, because they have delineated themselves, I think the content of the subject has delineated and entered into the approximation of causes. So that I'll just a little bit, I'll discuss. And you know that when you are talking about nutrition, about medical sciences and medical biochemical applications or whatever it is, it deals with the human body. And the human body has an exquisite design. I think all of us will agree. It is a designing marvel, but there are thousands of flaws and fragilities. These thousands of flaws and fragilities that make us vulnerable. If anybody is interested, can refer to two very famous books, The Man, the Mechanical Misfit. It was a 1941 old book, George S. Brooks. And another book I like to refer to the student friends, The Scars of Evolution by E. Morgan. Now, can we, look into the subject as a nutrition or, food science? We know that, high fat diet causes heart disease. This is nothing new information. But the question is, why do we crave for fat food? Or if I put the question in a different perspective, why natural selection has not eliminated those genes that make us susceptible to this disease? Why do we crave foods that are bad for us, but have less desire for pure grains and vegetables? Why we do keep eating when we know we are too fat? Why is our willpower weak to restrain our desires? So, like the examples are paradoxical. So we must discover the causes. And the cause, it is, so most of the disciplines are addressing to the proximate causes that answers what and how. Very few areas try to look into the evolutionary cause of why. That means we are denying that we are also the product of evolution. So we want to know that why some body parts are prone to failure. Like, I'll take one or two examples. Say for morning sickness, during early pregnancy, you have a nausea, vomiting, food aversion. Now the question is, why this aversion towards food? Is it evolved to protect the developing fetus from toxins? So these had to be answered in a larger perspective. And then another is the iron story. So likewise in nutrition, you have thousands of like these examples, paradoxical situations. And knowingly or unknowingly, the physicians if they are unaware, is going to treat those symptoms. But not even realizing that this has evolved in million years of time. Say for patient with chronic tuberculosis, automatically has a low level of iron. And if a dietitian or nutritionist or in a physician supplements his diet with iron, obviously what will happen? The infection will worsen rather than helping him out of the anemia. So modern dietary inadequacies are plenty, like vitamin C problem, because we all realize as a student of biology that scurvy is a disease of civilization. And there are other areas like modern nutritional excesses. What excesses that we are taking? Fat, sugar, and salt. But if you look into the evolutionary aspects, when human evolved into the areas of African savanna, it was in totally short supply of these three elements. Now what is happening? Every household is stocked with fat, sugar, and salt. So it is easily available to us. So the mismatches between the taste evolved from Stone Age conditions and their likely effects. So we have to understand the evolution of human nutrition. Now, as I was looking into the search engine and I put into the Google search engine, and I found that Paleolithic diet, I think those who are expert in nutrition, they would be able to tell us. The Paleolithic diet, the word was the most searched word in a particular frame of time. And now, if you look into the MBO, that is the European journal in 2008, it very nicely sums up that we are what we eat. And eating a healthy diet does not come naturally in the modern environment. So there are diseases of affluency. So there are diseases of poverty. That nutrition biology, the food science has to take care and come up with some solutions. Because we know that all these diseases, they do not result from random or malevolent forces. They all arise ultimately from the past natural selection. So when we look into the, what is the take-home message to all my students, friends, that when you are looking at a subject in a perspective, don't look at only the proximate causes and reasons. Try to look into the long history that evolved and made us human, whether we go for a protein-rich food, whether we go for starchy food, or whether it is the combination of both, and how it fits. And we should look into the cooking aspect. And that is very nicely put into one editorial column, that cooking is a signature feature of Schubert diet. But till date, we don't have a calorie counting system, the effects of cooking. Either we are detoxifying the toxin, or we are losing some important vitamins or some supplements. So we have to make it in a nutritional unique and come up with some solution that evolves and humans, they rise ultimately from these situations of mismatches. Lastly, I'll quote with a very famous saying, I often use it in my class, of a very famous evolutionary biologist. Nothing in biology makes sense except in the light of evolution, whether it is basic sciences, whether it is applied sciences. Thank you very much. Thank you, the college authority, Professor Pan and his whole team, for giving me this opportunity of sharing my thoughts. I think Sumedha is also here, so I should take a leave now because she is there. So thank you very much.
Thank you, sir. Thank you for your valuable words. Now I am requesting my colleagues, Dr. Saurabh Sarkar, please start the technical session. Our honorable speaker has already joined. Over to you, Saurabh.
সৌরভ, আমি তাহলে একটু লিভ করছি, জাস্ট সৌরভ।
হ্যাঁ স্যার, ইয়েস স্যার,
সুমেধা পরে কথা হবে, টাটা। আচ্ছা, সুকান্ত,
হ্যাঁ স্যার, হ্যাঁ স্যার, ঠিক আছে,
আমার একটা মিটিং রিশিডিউল করা আছে।
ঠিক আছে স্যার।
Thank you very much. Respected Principal Sir, Dr. Sopon Kumar Pan, Principal Guskora Mahavidyalaya, our honorable guest, Professor Dr. Obhijit Mojumdar, Professor, Department of Zoology, the University of Burdwan, invited delegates and dignitaries, eminent faculty members, and my dear student friends. At first, I would like to welcome all of you from entire organizing committee of International Lecture Series on Nutrition organized by IQAC and Department of Nutrition, Guskora Mahavidyalaya. I would like to now start our technical session with the permission of our respected principal sir. At first, our speaker of the technical session is Dr. Sumedha Roy. Allow me to introduce Dr. Sumedha Roy. Sumedha Roy, PhD, worked for her doctoral thesis work on molecular endocrinology at the Department of Animal Physiology, Bose Institute, Kolkata, with silkworm as model organism. Following this, she joined the Department of Zoology, the University of Burdwan, as an assistant professor and started exploring research areas related to fluoride and organophosphate pesticides, including induced morphological, neurological, behavioral, and reproductive toxicity in model organism Drosophila melanogaster. She served in Burdwan University from 2007 to 2019, nearly 12 consecutive years. Presently, she is working as a postdoctoral research scientist in the Department of Biomolecular Medicine, Faculty of Medicine and Health Sciences, Ghent University, Belgium. She is currently exploring different mutations in human RNF216 gene, which leads to a rare autosomal recessive neurodegenerative disease, Gordon-Holmes syndrome, demonstrating ataxia, dementia, and hypogonadotrophic hypogonadism. She has published more than 50 scientific papers in national and international journals and has successfully supervised 5 PhD students for their doctoral thesis. Thank you, ma'am, for joining with us. Now, over to you, ma'am. The screen is yours.
Thank you, Shora, for introducing me with those kind words. And thank you, the organizers of Guskora Mahavidyalaya, for giving me this opportunity to share my little knowledge with you, the student friends. And since it is regarding nutrition, the lecture series is a very interesting one. And as rightly pointed by the keynote speaker, Dr. Obhijit Mojumdar, more fondly I call him Obhijit Da, he has rightly pointed that maybe sometimes it is more necessary to know what we eat and how we eat. Our diet is really important. And since Saurabh has already talked about that, I have been working with toxicology as a background. So I will be talking a little bit on the nutrition and toxicology interface. So now I share my screen. I think you should give me the rights to share the screen, because I cannot share the screen now. So can you please, the technical people behind this thing, can you please give me the rights to share the screen? Hello, I cannot share my screen now, so can you please help me sharing the screen? I'll request Shukendu or Saurabh to just find out if I can.
Sir, ma'am.
Okay,
wait
okay,
a-
yeah, I now I can make out, yeah. I think I can, my screen should be visible by now. Is it visible? Saurabh, can you please tell me?
Yes, yes, ma'am.
Okay, fine. Thank you for being actually fumbling with the thing. So I don't know why this is coming up. Why is it mirroring? I don't know. There is a problem.
Ma'am, actually, after presenting the screen, you please go to the screen, ma'am.
Yeah,
You
I'm
already
going to that screen.
Already showed that.
Yeah, it should be there.
Yes, ma'am. Visible, ma'am. Visible, ma'am.
Yeah, I can see, but I just want to go to the presentation mode, since it is, you just see, yeah, now I can, I think it
Yes,
should
ma'am.
be.
It is visible now.
Yeah, now I think it is visible. Okay. No, because I just wanted to shift to the presentation mode. Since I was talking about nutrition and toxicology interface, so coming into the points regarding nutrition toxicology. So nutritional toxicology is actually the subject which combines the research approaches of nutrition and toxicology together. So many problems of substantial importance to health and food safety is actually, it should be given importance, and their interactions in the nutritional process, this actually requires a very good study. It is actually a very good study material, and the relationships are very vital. So the relationship between nutrition and toxicology may be described in three basic categories. So what is the influence of nutrition on toxicities? What is the influence of toxicants on nutrition? And if the nutrients are toxic by any means or not, that we also should know. So these relationships should be studied properly and understood properly, so as to guide a person properly to the nutritional requirements. And so I must say that, taking into consideration these three categories, what I have already mentioned, that the nutrition of the whether the nutrition has got any influence on toxicities, or whether the toxicants has any influence on nutrition. So I'll just give few examples to make it a little bit clear for the student friends present in this session. Now, talking about trypsin inhibitor, which is very commonly found in soyabeans, the whole soyabeans that we consume very frequently. These whole soyabeans are known to have the trypsin inhibitors. So trypsin inhibitors are the chemicals or certain enzymes which are actually anti-nutritional in nature. So if it is anti-nutritional, how is it anti-nutritional? Because it actually resists the breakdown or proper metabolism of the protein in the small intestine because of the presence of this trypsin inhibitor. So excess consumption of soybeans is actually questionable whether it is good or not. So in that way, a food can be anti-nutritional also. In the same way, if we talk about acetaminophen, which is a common anti-inflammatory drug, it is just like paracetamol. Some people, they actually have to take acetaminophen. And this drug, for example, it actually affects, means when the drug is taken in excess dose, it has to be detoxified by the liver system. And when the liver detoxifies or actually gets rid of the excess drug from the body, in the process, it also needs glutathione and some sulfate for the process. So there should be enough sulfate amino acids present in our body so as to get rid of excess acetaminophen. But at the same time, while getting rid of acetaminophen, the body also siphons out cysteine. And thereby, cysteine is less available in the body when we consume more acetaminophen. So when cysteine is actually getting out of the body, it is less available for the skeletal muscles, and thereby it has a very detrimental effect on the skeletal muscles if we consume acetaminophen in excess dose. So this is another example of a proper drug, which is a very common drug. Its overuse or excess use or continuous use might be actually playing a trick with the nutritional status of our body and thereby playing a detrimental effect on the body as well. And another interesting fact I just want to share, which makes it more easy to understand. We know that cadmium is a toxic metal. So any heavy metal is generally, if we come in contact with a heavy metal, it is going to show some adverse effects on the body. So this cadmium, how is it absorbed? When it is absorbed in the body, it has been noted that if the person, if a human being has dietary deficiency of calcium or iron and low in protein content. So a person, if I am already deficient with calcium and iron, I will be more prone to absorb more cadmium in the body. And it has been shown through various experiments that a person with low ferritin level have twice the normal rate of oral cadmium absorption. So that's here I must say that the women who are of the childbearing age, who has a constant problem of iron deficiency in their body, they have a greater chance of having more cadmium absorption in the body, thereby more chances of having toxicity, cadmium-induced toxicity. So here is also an example where we see that the dietary constituents of the person, maybe more calcium and more iron in the body, might be helpful to resist cadmium toxicities. So this is what I should be pointing out to the fact that when we are talking about these three categories of influence, we can also take them as examples. Now coming to poison, as we are talking about toxicity, firstly, what is a poison? So interestingly, 500 years ago, a Swiss physician and chemist, he pointed out the fact that all things are poison and nothing without poison. So what determines its poisonous nature is the dose. So solely the dose determines that a thing is not a poison. So anything in excess can be poisonous. That has been pointed out maybe 500 years ago. And now, while we are talking about toxicology, we should be, already it has been pointed out by the keynote speaker and also by Professor Pan, that modern toxicology is actually a combinational subject, where it goes beyond the study of only the adverse effects of exogenous agent. So when we are talking about toxicology, it is not only that what is coming outside from the body and it enters the body and it creates some adverse effect. That study is not toxicology alone. Toxicology is much more than that. Actually, nowadays, many of the toxicologists are busy in studying mechanisms where endogenous compounds, means the compounds that are formed within our body, how those compounds can be detrimental, how those compounds can be adverse for our body. So nowadays, more and more people are into research with oxygen radicals and other reactive intermediates that are generated from the xenobiotics or endobiotics. So it's more within, the poison within should be studied also and should be considered a part of toxicology. And this is all about history. I think we have already heard about Hippocrates, who has added a list of poisons and clinical toxicology principles. And so it is as old as the Ebers Papyrus in 1500 BC that has already talked about many of the toxicants like the aconites, opium, metals, antimony, and all these things, because previously it was more that people thought toxicology is all about warfare. It is all about killing, or it is just how you can get rid of the, maybe in the war, how you get rid of your enemy is through the toxicological or toxicants used. But nowadays it is more that the enemy within should be studied more clearly to understand how we address the problem of toxicosis in our body. So now, talking about toxins around us, we all know that if we eat a wrong mushroom, or if we eat too much of pufferfish, or if I'm bitten by a snake, this is all about toxins. So there are a list of toxins which are available. Now, starting with the pufferfish, we know that pufferfish contain tetrodotoxin. Tetrodotoxin is actually a toxin which is nearly 1200 times more poisonous than cyanide. So it can kill humans in no time, I must say. And tetrodotoxin actually interferes with the voltage-gated sodium channel in the nervous system. So nerve cell voltage-gated sodium channels are interrupted or blocked by tetrodotoxin. Thereby, it is easily, you can understand that it is a neurotoxin which we are talking about. And about mushroom, we all know some mushrooms are really poisonous and we should be very careful in eating mushrooms. And what about snake? We all know that snakes are venomous and sometimes they create havoc when they are in farmers, in case of when they are in the field. Now, coming to the poison, so what we told that anything which is actually taken in greater dose can be poisonous. So any substance, when it is taken in the system, it causes some injurious effect, and that injurious effect is not only mechanical or thermal, but it is metabolic effect that we see in case of a poison, because the metabolic effects actually causes detriment to the health. So the adversely affected health condition arises due to intake of poison. Now, the poison can be a toxicant, that is, it can be a chemical of chemical origin, or it is coming from outside, that is exogenous source. And when we are talking about toxins, it can be from the biological source, so toxicants and toxin. And there are different poisons available around us. So talking about poisonous marine creatures, poisonous fish, venomous snakes, ticks, scorpions, spiders, poisonous plants. And interestingly, I think some students will find it very interesting that several birds have toxic feathers. So when a person touches the feather, then the toxic impact starts. So when it is touched or it is ingested, there are toxic reactions, and several mammals are also poisonous. Now coming to a common, it's actually when we see some grains or corns, we come across this Aspergillus flavus. So Aspergillus flavus is actually the type of fungus which actually has the toxin known as the aflatoxin. So aflatoxin is responsible for causing aspergillosis. And aspergillosis is when we consume the corns or peanuts which has this fungus growing on it. And it is very common when the grains or the nuts are stored in a moist condition. This kind of fungus is very common throughout the world, and this aflatoxin causes irritation in the respiratory system and also to the eyes and ears. And sometimes if consumed in excess, there is a chance of liver cancer also, because liver is the most important organ of the body which actually participates in the toxic metabolism of any chemical which enters into our body. Our liver tries to get rid of that chemical. So there is always a pressure on the liver, and excess consumption of any toxicant will obviously lead to liver ailment and might also lead to liver cancer if longer exposure occurs. Now, taking alcohol, just to understand the pharmacology and the toxicology, that how an absorption takes place and how far the concentration of the ingested material shows the impact on the body. So to take that as an example, I'm considering alcohol toxicity, because I think all the audience is quite aware of the detrimental effect of alcohol consumption in excess. So when we are talking about pharmacology and toxicology of alcohol, as an example, firstly, what comes into play is the absorption of alcohol. And alcohol is, interestingly, it is absorbed by simple diffusion process, and 25% of ingested of alcohol, in 25% of the dose of the ingested alcohol is absorbed in the stomach, and the rest 75% of the ingested dose of alcohol is absorbed in the small intestine. Now, alcohol concentration in the ingested beverage, it has been seen that optimal absorption occurs for alcohol when it is within the range between 10% alcohol volume by volume to 30% alcohol volume by volume. But excess to 30% alcohol causes irritation in the gastric mucosa and increase the mucus production in the gastric region. So now, what is the effect or on the absorption? What are the factors that affect? First of all, the presence of food in the stomach. If there is food in the stomach, then it will prolong the gastric emptying time, resulting in lower or delayed peak of alcohol absorption in the stomach. So, and the physiology that we are talking about is that when there is alcohol in the stomach, there is the blood supply to the GI tract, gastrointestinal tract is changed, and there is the motility is also altered, and that actually alters the speed with which the alcohol enters the small intestine. And we have already seen that 75% of the alcohol dose is actually absorbed through the small intestine. So if the motility is lowered, then there is an effect on the absorption. And talking about alcohol metabolism. So we know that 5 to 10% of the ingested alcohol is excreted. And there is biotransformation of the ethanol that takes place in the liver. So as soon as a person consumes ethanol, it is liver which comes into action, and it tries to biotransform that ethanol. And in the process, there is an enzyme which is known as the alcohol dehydrogenase, the enzyme which is very important. And so I just use maybe a pointer. Okay. So when we are talking about alcohol dehydrogenase, the enzyme which is actually very relevant, and it transforms the alcohol into toxic compound, the acetaldehyde. And this, if in excess, if it accumulates in the body, it's a carcinogen. So we are talking about alcohol, and when it gets transformed in the body, we should also be knowing about the 5 to 10% of alcohol and how the body eliminates it. So elimination of alcohol from the blood, it actually follows a zero order kinetics. So zero order kinetics is actually the kinetics by which follows that whenever something toxic gets into our body decides to throw away or eliminate actually the constant amount of that substance per unit time. So in case of zero order kinetics, which follows when a person eliminates constant amount of the toxicant out of the body per unit time. And there is another term which we might come across in case of other toxicants, which follows first order kinetics. In case of first order kinetics, it is the constant concentration of the toxicant that is eliminated from the body per unit time. So if we consume more and more of a toxicant substance, then it will eliminate a constant percentage of that toxicant. So it will be more amount in that case in the first order. But in case of zero order kinetics, it is always the constant amount which is eliminated. And it ranges from 10 to 20 milligram per 100 ml per hour. So no matter how much we consume, how much alcohol a person consumes, the body can eliminate only 10 to 20 milligram per 100 ml of the alcohol in an hour. So that is the rate of consumption, and the amount of consumption is really vital for toxicosis related to alcohol. So if a person consumes alcohol, but the person actually consumes more alcohol with a longer hour of exposure, then the person might be eliminating the alcohol in that way better than a person who consumes a whole lot of alcohol in a very small duration, because that person might not be able to eliminate a greater amount of the alcohol out of the body, because we know that alcohol follows zero order kinetics. So the average rate of elimination is 15 milligram per 100 ml per hour. Now, I think this part of the panel needs no description or needs no explanation, because we all know the effects of alcohol on the body. There is vasodilation, there is a feeling of warmth when alcohol is consumed. And we have seen that people who consumes more alcohol, they start talking more. So there is more talkativeness or more euphoria, which actually the person gets rid of any inhibition. And But at the same time, excess of the alcohol consumed can be acting as a nervous system depressant and the brain and spinal cord depressant, and thereby the person might feel sleepy or sedated when excess of the alcohol is consumed. So, and alcohol and death, primarily the mechanism of death due to alcohol consumption is intoxication of the respiratory depression. And when we talk about BAC, means the blood alcohol concentration, we have seen that nearly 350 milligram per 100 ml of alcohol in the blood might be responsible for respiratory paralysis, and that might lead to death. Now, coming to another part of toxicology. So until now, we were talking about toxicants that are actually we can consume, but there is another field of toxicology which is also interesting, and that is genetic toxicology, where some agents actually can cause change in the genetic material, that they might induce some genetic damage and leading to toxicosis. And the agents responsible for genetic toxicology might be pharmaceuticals, consumer products, chemicals, radiation, and viruses. So these agents, somehow, if they actually come in contact with our genetic material, in case of humans, it's DNA, we have seen that it causes genetic changes leading to toxicosis, and thereby it might be the toxicosis in case of genetic toxicology might be better termed as carcinogenesis. So why is genetic toxicology important? Because genotoxic and carcinogenic agents are ubiquitous, because wherever we go, we might be exposed to radiation, which is actually sometimes genotoxic, more or less genotoxic, the cosmic radiations, and genetic damage can occur constantly, and it is occurring, and our DNA has got a repair system. So most of the time our DNA can rectify the damage which is done. It is trying to, but some damages are irreparable and irreversible, and there comes the problem. So now talking about some food items which are also very not so good for health, but we are sometimes pretty happy to consume a cup of coffee. A cup of coffee is not a problem, but excess of the chemical, if it gets inside our body, might be problematic. And there is another common thing that is known as Brussels sprout. Since I'm from Belgium, Brussels, and just talking from Brussels sprouts are very common in Europe, and these sprouts they look very similar to small cabbage, and they, when consumed excess, might cause to excess accumulation of allyl isothiocyanate, and allyl isothiocyanate is weak carcinogen, and excess consumption of that might lead to accumulation of that isothiocyanate in the body and might lead to carcinogenesis if consumed in super excess. It's not that if you consume one helping of Brussels sprout, you are going to get cancer. It's not that. And talking about coffee and other chemicals related to it, we should be talking about the cigarette smoke, which contains benzoalpha-pyrene. And we all know that benzoalpha-pyrene, it actually forms an adduct in the DNA. So it binds to the DNA and it forms adduct and causes damage in the DNA, which might lead to carcinogenesis in the long run. So genetic damage is actually tied up to multi-step process of carcinogenesis, because genetic damage once is one hit, means you just give a hit to the DNA, and further of the same damage, if it goes on and on for longer years, then that hit or that blow to the DNA might be a cause of concern and might lead to cancer in the long run. So carcinogenic effects might not be very evident for many years. It might only be evident when there is already proliferation in the cells leading to focal lesion and thereby forming tumor in the long run. So genetic toxicology is another branch of toxicology or subdiscipline of toxicology which needs attention. So there are more subdisciplines of clinical toxicology, which is neurotoxicology, immunotoxicology, developmental toxicology, embryotoxicity and phytotoxicity, then reproductive toxicology. Actually, from the names, I think the students might be easily understanding when we are talking about neurotoxicology, we are talking about toxicology to the nervous system, leading in the nervous system, toxicity which is actually concerned with the neurons or nerve cells. Immunotoxicology leads the problems in the immune cells, and the developmental toxicology when there is a developmental error coming out of excess or of any toxicant in our body. Embryotoxicity or phytotoxicity is also common. We know that the fetus is well protected, but still there are some toxins which do play a havoc for this embryo or the fetus. And reproductive toxicology is that branch of toxicology which deals with certain changes in the reproductive cells, in the reproductive system that might lead to sterility or might lead to low productivity of the organism. So different forms of toxic agents are there, metals, chemical agents, biological agents. Now coming to, I'm just taking one example of a chemical agent or chemical toxicant, and I'll just explain how nutrition and chemical toxicant can be interrelated through one of the experiments that we had done a few years back in Burdwan University when I was associated with Burdwan University. And I'm thankful to the students who have been associated with the research. And so here, what we are talking about, what I'm going to talk about is a chemical toxicant, the pesticide. Now, we know what are pesticides. Pesticides are substances that kill or repel harmful pests from the agricultural fields. And we know that we, the human beings, when we consume certain vegetables, unintentionally also, we are getting exposed to these pesticides. So we are the non-target organisms. The target organisms are the pests, the harmful pests, maybe the rats sometimes, and the insect pests that are playing a detrimental effect for the agricultural fields. But we, the people who consume the agricultural products, are also the non-target organisms. And what we wanted to unravel was to see if the pesticide, what is the effect of the pesticide on non-target organism, and how a nutritional, or what I must say, a nutraceutical can help in that process. So taking from the different categories of pesticides, I have highlighted organophosphates because this experimental work was actually designed with an organophosphate. We know that organophosphates are a major group of pesticides, and it actually works through blocking of the acetylcholinesterase. It actually blocks the acetylcholinesterase enzyme. It is in the synaptic region. So from the synaptic region, from the mention of the synaptic region, you can understand that it is a neurotoxin, because it actually creates havoc in the nervous system. And so the organism, that is the pest, dies soon after the application of the organophosphate. But chronic exposure to organophosphate in case of non-target organisms have been reported to cause fatigue, memory loss, weakness. There is joint and muscle pain, and depression also in humans. So when there is chronic exposure to organophosphate in low dose, it might lead to so many symptoms. And acute poisoning has been associated with nausea, vomiting, abdominal pain. Now, coming to the type of organophosphate that was used for this research was acephate, which is a systemic pesticide. It also is acetylcholinesterase blocker. And it has been used, it has been in use, and now also it is being used in fields of tomatoes, potatoes, lettuce, carrot. So it is actually used against the Lepidopteran pestes. Lepidoptera is, I think you must be knowing, the moths and butterflies. So moths are, for Lepidopteran pestes, there are some paste which actually has affinity for tomatoes and potatoes and lettuce and carrots. Against them, this pesticide has been in use. And here I must say that in this experiment, we tried to find out if vitamin C, that is L-ascorbic acid, can be used to be a compound or a nutraceutical against the toxicity induced by organophosphate. So we tried to explore that if there is any positive role of L-ascorbic acid, that is the vitamin C, against the toxicity that is induced by the organophosphate acephate. And in the process, we actually were working on Drosophila melanogaster as one of the non-target organism for the toxicological experiments, because time and again it has been seen that Drosophila melanogaster, being a very good model organism, it can be used in toxicological evaluations also, and because of its short life cycle, high fecundity and sexual dimorphism, and not only that, it shares 50% genetic and physiological homologies with the higher vertebrates. So that makes it more important or more interesting as a model organism for toxicological evaluations. Now, so the experimental objective was to see if there is any value of the nutraceuticals in the diet. So we were exploring effects of acephate in the non-target organism. Along with it, we wanted to explore if there is any protective role of L-ascorbic acid, that is vitamin C, in the diet against acephate-induced toxicity. So this was just to simplify. I just gave this cartoon. And what was our experimental design was that we had first instar larvae in culture of Drosophila melanogaster. And in the first side, in the left side of the panel, you see that it is control food. So it was only food that is the control food, that the standard Drosophila medium in which the first instar larvae were maintained and they were grown in it. But the second category was the category where acephate was used at a particular concentration. So as to see if there is any toxicity in the flies or in the larvae which were reared with food containing acephate. And the third category was a category which was of our interest, that we gave the same concentration of acephate in the food, but we added different concentrations of vitamin C, so as to see if this L-ascorbic acid has got any role to play or to reduce the acephate-induced toxicity in the flies. So we have collected the third instar larvae, we have done several assays, and these are the treatment categories that have been used. So just mind it when we are talking about the category 1, we are talking about control, that is Drosophila that were reared with standard Drosophila medium, that is without acephate or without vitamin C. And the number 2 category was with acephate, that is a particular concentration of acephate only. And number 3, 4, and 5 were the groups where we have used the acephate, that is the same concentration of acephate, and increasing concentration of vitamin C, 25, 50, and 100 microgram of vitamin C in these categories. And we have carried out several assays. Firstly, climbing assay. Then we actually took the help of scanning electron microscopy to see the compound eyes of the flies, if there is any developmental change in the flies because of the toxicant. And we did acridine orange staining for apoptosis detection in the eye imaginal disc. We also did the MTT assay, which is actually to see if how many cells are dying in the fat body or in the brain ganglia in case of the treated larvae. And then we have done some of the enzymes. The enzymes, firstly, I should just explain one thing before proceeding to the next slide, the climbing assay. Climbing assay is actually the assay by which we determine the climbing ability of the fly. So we take a particular container which is marked at different areas, and we let the flies with a stopwatch, we just see that how many flies can actually run or climb the 5 centimeter line, and how many flies remain below the 5 centimeter line within a 30-second time interval. And we had to count these flies if there is any change in the motility of the flies because of the organophosphate exposure. Because we have seen that, we have already known that the organophosphate is acetylcholinesterase blocker. So there must be certain change in the motility because the nervous system gets affected. So there is a change there might be a change in the motility because of toxic reactions. And I have already talked about the scanning electron microscopy, acridine orange staining, and MTT assay. Apart from that, we have carried out certain enzymatic assays with the tissue homogenates that were prepared from these categories of the different categories of insects. And in the process, we have carried out certain enzymes, that is superoxide dismutase, glutathione transferase, cytochrome P450 assay. So these assays were actually meant to understand the toxic status of the organism. So if the body is actually under the threat of toxicosis, the body will try to eliminate the toxicant out of the body. And the organism in that process will actually increase the activity of certain enzymes, which help the organism to get rid of the toxicant. So these enzymes were assayed just to understand the toxic status of the body. Now, the statistical analysis were done, and I'm just going to present you the results. So we were counting the flies that were above 5 centimeter. So and some flies which were below the 5 centimeter mark, and 1, 2, 3, 4, 5 are the different categories of treatment. So the first category was the standard or the control, where no acephate, means no organophosphate, no vitamin was given. So the flies were, all the flies, pretty high number of flies were above the 5 centimeter mark because they were able to climb, they were fit and agile. But when we fed the flies with organophosphate, we saw a decline in their mobility, and we saw that most of the flies, they failed to climb. But when we started giving vitamin along with the acephate or the organophosphate, we see that there is a rescue in the process, and we see that in the third position, it has been rescued. So along with organophosphate, when the flies consume vitamin C at a particular concentration, the flies are able to be fit and agile as that of the control ones. So similarly, the lower part of the graph represents the number of flies that were below. So in case of the acephate treated flies, most number of flies were below, but the flies were able to climb high. So the number reduced in case of below 5 centimeter mark. Now coming to the acridine orange staining. So this is the eye imaginal disc. So in case of larva of the Drosophila melanogaster, there are a bunch of cells which are destined to produce a particular organ. So the cells which are destined to produce the eye are known as the eye imaginal disc. So a particular kind of staining, which is with acridine orange, and observation under fluorescent microscope helped us to understand that in the control flies, the eye imaginal disc didn't show up any fluorescent dots. Fluorescent dots are actually the sites of apoptosis. So when the cells die, they cause this fluorescent to appear. So in case of the number 2 category, we see that quite a few, many cells are there which are actually showing the apoptosis. But as we go higher up, though I must say in the 4th category, the rescue was not that good in case of apoptosis. But when we reached the maximum concentration of vitamin, that is 100 microgram, along with the acifit, we saw that the eye imaginal disc is somehow, this part of the eye imaginal disc is rescued. And what we see in the scanning electron microscopic figures was that the control flies, they show the eyes, they were very nice and regular. But with acifit exposure, we see that there are grooves and ridges which appear in the eyes of the flies that are exposed to organophosphate. But when we have treated with organophosphate as well as vitamin C, the eyes get bettered. And in the 5th category of treatment, we see the eyes have been rescued. And typically, there is no phenotypic expression of the grooves and ridges in case of the 5th treatment category. So along with that, we have we wanted to explore how many cells die in the brain or in the fat body, because we have taken out brain ganglia and we have counted the number of dead cells through the MTT assay. And we saw that in the 1st category, most of the cells were living. So there was, in the treatment categories, the MTT assay showed that most of the cells were living in case of the control, but most of the cells were actually getting affected when it was with the organophosphate. And when we applied vitamin C along with it in the diet, we see that the cells are being rescued. So we get to have few but significant number of living cells in the brain as well as in the fat bodies through MTT assay. And talking about the different enzymes, that is catalase, superoxide dismutase, glutathione transferase, we have been able to show that the body wants to eliminate the toxin out of the body. So these enzymes will actually show you the toxic status of an organism if it is exposed to a toxicant. So here we know that the 1st category is control. So there was no toxicant exposure. So the organisms, they showed a enzyme level of this one, but there was a significant rise in SOD when it was exposed to acifet. And we see that with vitamin C, that exposure of acifet, the effect of exposure of acifet has been reduced because vitamin C has tried to rescue the organisms out of the toxic response. So in the glutathione, in the catalase, we see a very good response. The catalase comes down to nearly normal control level in when we apply vitamin C along with the acifet. And the cytochrome P450 is also induced when there is organophosphate exposure in the category 2 of treatment, but category 3, 4, and 5 do show a reduction in the cytochrome P450 activity. Thereby, we get a confirmation that the body, that the toxic status of the body is altered or changed due to addition of the nutraceutical in the diet. So the toxic status is bettered. We are actually moving away from the toxic response in the organism if we have the vitamin in the diet. And along with it, we also assayed what is the acetylcholinesterase, because we were talking that about acetylcholinesterase inhibitor. Organophosphates are acetylcholinesterase inhibitors. So when we actually assay the acetylcholinesterase, we see that in the control, acetylcholinesterase activity is nice and high, and it gets reduced when there is the acifet or the organophosphate exposure. But when we add vitamin to the diet with the organophosphate, we see that it comes up to nearly normal level of acetylcholinesterase activity. So thereby we are able to prove that the vitamin in the diet does play a role. Apart from that, we also carried out the COMET assay, where we just wanted to see if there is any DNA damage occurring in case of cells that are exposed to the acifet or the organophosphate. And it has been done with larval hemocytes, the blood cells. So these are the immune cells also of the larvae. And we see that in case of the exposed, acifet exposed cells, there is a COMET tail. That is, the degradation of DNA actually causes this COMET tail. So appearance of a COMET tail confirms that due to exposure to the organophosphate, there is degradation in the DNA taking place, whereas the cells which are already exposed to acifet but are at the same time exposed to vitamin C, there is a reduction in the COMET tail, and that is sufficient to understand that the DNA damage is minimized. And this is a cartoon which represents the same thing that I was just talking in the previous slide, that there is the tail length is increased in case of the acifet or the organophosphate exposed cells, but the tail length of the DNA, that is the fragmentation of the DNA, is reduced. So DNA damage is reduced, and it is coming down to the normal level in case of the organophosphate as well as vitamin treated categories. So from here we understand that acifet has already reduced the locomotory activity, because we have seen that the climbing activity was affected when it was exposed to organophosphate. So it is the neurotoxic potential of the organophosphate that actually disrupts the coordination between the muscular activity and the nervous system, thereby affecting the locomotor ability. And not only that, the acifet also generates the reactive oxygen radicals, because we have seen it in the different enzymes. The superoxide dismutase enzyme assay has confirmed that there was a release of reactive oxygen species, which was actually the body was trying to get rid of the reactive oxygen radicals, and thereby was increasing the activity of superoxide dismutase enzyme. And in the process, there was also the DNA breaks that were confirmed through the COMET assay. There was the intrinsic antioxidant enzymes, which were also showing high activity when we have exposed the organisms to organophosphates alone. But at the same time, in all these categories, we saw that with addition of L-ascorbic acid or vitamin C as a reducing agent, it converts the free radicals into stable, harmless molecules, and it terminates the oxidative damage and actually helps the organism to come out of the toxicity, toxic status or the toxic responses. So this is what I was talking about, the oxidative stress that was induced by the acifet, and the vitamin C actually helps or bars this oxidative stress and in a process, it will actually rescue the organism from all these parameters. And when we add vitamin C to the diet, it helps the organism to get rid of the toxic responses. So this was the article that was published from our group, which we were working in Burdwan University. And I'm very thankful to all my students who have participated in this research. And so to summarize the fact, I must say that food can act as a source of nutrient and can also act as a source of toxicant. So we need to understand the influence of nutrients for food safety and toxicity, and we should explore the influence of toxicants on nutrients, and we should also understand nutritional requirements versus toxicity, because the experiment that I was just explaining a few moments back was that when we are prone to exposure to any sort of toxic agent, then we should be planning our diet in a more rational and more intelligent way, that our diet might be helping us against the toxicity. So there is a relation between the nutrition and the environmental toxicants, and we should also understand and unravel the benefits versus toxicity of the phytochemicals, because time and again we are fond of using certain phytochemicals, but we should be aware of the toxicity of those phytochemicals and the dosage of usage also in the diet. So there are certain regulatory issues in establishing the food safety and standards, and those regulatory issues are taken care of by different agencies which take care about the food safety. And I must say that EFSA, the European Food Safety Authority, takes care of the food safety in the European Union. And there is the Food and Drug Administration, that is the FDA in USA, and also the Food Safety and Inspection Service, that is known as FSIS, which is a public health agency in USA, which takes care of the food safety. And in India, we have the food safety standards, which actually takes care of the food safety in our country. So, and apart from these few, there are several food safety agencies who are working on day in and day out to understand the safety measures that should be taken for certain food and consumer products. So we must be careful about what we eat. So as rightly pointed and said by Dr. Mojumdar, that we are what we eat. So be rational, be intelligent in choosing that. And I must complete my lecture. I must just come to an end with the words of Anne Wigmore. She was a Lithuanian-American holistic health practitioner. She was a passionate teacher and a researcher, and rightly said that the food you eat can be either the safest and the most powerful form of medicine or the slowest form of poison. So we should be careful about what we eat and how we eat. And thank you to all my students from whom I learned a lot, and I think I'm still learning. And thank you all for your patience and attention. Thank you.
Thank you very much, madam, for your nice presentation. We are really fortunate to have you in our international lecture series program, and definitely enriched by your fantastic lecture in lucid language. Now, we would like to pick up some selected question and put forward in front of you. Please allow us.
Yeah, sure.
First question, Mousumi Pal, how do we get rid of toxic elements from our body?
So I think if you have, can you hear me, Saurabh?
Yes, ma'am, definitely.
Okay. So I think in the last part of my lecture, since because it was a time schedule, I was going beyond it, so I tried to minimize the thing. But in the experiment that I was presenting, I have talked about several enzymes. So in the body, we do have some detoxifying enzyme system working for us. So whenever a toxicant is entering in our body, those enzyme systems are taking care of how we are going to get rid of that toxicant. And in the process, the liver is an organ which takes part very, means it's the first and foremost organ that takes part in the toxic removal, or the removal of the toxicants from the body. And I have already mentioned about cytochrome P450. I think you have already come across the names that I have mentioned, because when there is oxidative stress, we see that there is an increase in the superoxide dismutase activity. There is LPO, lipid peroxidase, and there's glutathione transferase. All these enzymes are actually the relevant enzymes which take care of when any toxicant enters our body, and just to get rid of them.
Thank you, ma'am. The next question is from Nasreen Rahmana. What is oxidative stress?
Yeah. So oxidative stress is actually when our body, in the process of metabolism, releases free radicals of oxygen, and that free radical or excess free oxygen is harmful. So first, when I started my lecture, I told you that anything can be poisonous, and that is the dose determines the toxicity of any chemical. Similarly, excess free radical, excess oxygen, free oxygen in our body is detrimental and harmful, and is considered as the agent causing the oxidative stress.
Next question is from Deep Chattaraj. Are berry and aloe vera help to detox liver? If so, how do they do so?
Many phytochemicals are actually very good for liver, but I am not very much aware of the chemical constituents of the berry or aloe vera, since I am not very well versed with the chemical constituents. But yeah, obviously there are certain agents which are good for releasing out excess toxin from our body. I must suggest only one thing that is always available and is good is water. Drink a lot of water and healthy diet, not excess fat. And yeah, obviously the fruits are good for effect, but only be aware if the foods should not be contaminated with pesticides. But I am not very sure about aloe vera and berries. Sorry.
Thank you very much, madam, for your nice deliberation. We are really honored and grateful to you for joining with us in our joyful learning program. Thank you very much. Now,
thank you, and thank you to all for giving me this honor, and thank you, Shourabh, for nice words. Thank you, Shukendu. Okay, I'll just leave. Means I'll not leave. I'll be here, but I'll just switch off my audio. Okay.
Okay, ma'am.
Thank you. Our next resource person is Mr. Sobhyasachi Chakraborty, DABT. Let me introduce Mr. Sobhyasachi Chakraborty. Mr. Sobhyasachi Chakraborty is a senior consultant at Toxminds, Brussels, with more than 14 years of research and consulting experience in the human health hazard and risk assessment of chemical substances. He is experienced in the hazard assessment of chemical ingredients used in consumer products, including cosmetics and other personal care products. He has extensive experience in developing safety assessment of chemicals under the REACH regulation. He is also involved in the development of the chemical exposure and risk assessment outputs as required under the REACH, BPR, and agrochemicals regulations for human and environmental health using prediction and modeling tools. Over the last couple of years, Sobho has also gathered experience in working on interactive ingredients and impurity-related toxicological assessment for pharmaceutical products. He is a diplomat of American Board of Toxicology, that is DABT, with a master's degree in pharmacology from the Manipal Academy of Higher Education, India. Welcome, sir. Now the screen is yours.
Thank you, Saurav. And thank you to the organizers of this lecture series of nutrition from Gushkora Mahavidyalaya. I'm a pure Bengali, so I'll basically conduct this short discussion on the scientific writing in English as well as in Bengali, so that you understand the terminologies or how do we write in our daily scientific work. So I'll present my screen now. And do let me know when you see the screen. Can you see the screen?
No, sir, not still.
Can you see it now?
Yes, sir. Yes, sir.
All right. So I'll present it in a presenter mode. I hope you can see the screen.
Yes, sir.
All right. So thanks, Saurav, and thanks to the organizers again. It's been a privilege to talk with you and discuss with you on the way we write and the way we have learned over the last few years. All right, so this discussion will be on, in general, scientific writing, and more specifically to cater to the requirements of your syllabus, the review articles. Now, I must say that I am not an expert in basic research article or review article writing per se, because after my formal education in Manipal University, I directly joined the industry as a pharmacologist and toxicologist in Dr. Reddy's. And then I moved to another industry, and since then, over the last 14-15 years, I have been working as a consultant, mainly in regulatory toxicology field. Now, the way we work does not allow us to publish any research work or review work, because we normally work with client confidential data. So they are all confidential data directly from the industry, the plants, their products. Therefore, we are not allowed to publish. But what I can share with you all is how we write and how we have learned to write over the last few years. Of course, with guidance from my seniors, my immediate supervisors in different companies, as well as in my current company, ToxMinds. So today, the flow of the discussion would be to provide you first some basic differences between a research article and a review article. I'm sure you must have seen and read a lot of research articles as well as review articles, but just for recapitulation, I will again point out the basic differences. And then a few of the other items or the agenda in the discussion. For example, how do we plan? What are the things we remember when we write a review article? And then for the benefit of the students, what are the common pitfalls in scientific writing, and what are the language tips? So this will be a short discussion. It could be a very interactive. Now, coming to the opening slide, I found interesting statement by George Gopin and Judith Swan. It was way beyond 1990s when they have published some art of scientific writing, how to do. So as you see here in the screen, they say that the fundamental purpose of scientific discourse is nothing but can you put your audio on mute? Someone has turned on the audio button. I can hear my echo. All right. So as you can see in the screen, it says that the fundamental purpose of any scientific writing is not only the presentation, how do you present the information or your thoughts, but it's the accurate and actual communication. So the purpose is not to please the audience with your sentences or text, but it's the purpose that the majority of the audience could actually understand what you had in mind or what you are trying to write. So that's the actual purpose of any writing. So whatever you have conducted in your basic research, or let's say in a review article, whatever you are trying to present or write, it should accurately be perceived by majority of the audience. That's the purpose. Keep this in mind always. Now, there are some basic differences between a research article and a review article. Okay, so on the left-hand column is a research article, and on the right-hand column is the review article. Now, as you know very well, that for a research article, it's the viewpoint or the thoughts of the author who is publishing a research. It's directly coming from the original research done on a particular scientific question or query or problem. On the contrary, for a review article, when you do a review article, it's the collective views of different authors, but on a particular topic. So it's the discussion of the viewpoint of different authors. Again, coming to a research article, the research article is usually a new content, okay, based on your research. But for a review article, it's already published content which you are discussing. And a research article usually is mandated by a word limit, and each journal has its own word limit for different sections or for the article as a whole. And for a review article, it's usually shorter than a research article, although some of the journals might prescribe you a word limit. So these are the basic differences. Now, what I haven't written here, but would like to discuss with you, is that when you write a review article, it's a discussion, as I have pointed out in the first bullet point, it's a viewpoint of different authors. So without bias from your own prejudice or from your own viewpoint, a review article basically discusses the viewpoint of different authors. So when you start publishing or writing a review article, first of all, you have to read the different scientific discourses or writing from different authors. But also you have to understand their viewpoint. from where they're coming, what was the context from which they're coming, and what are they trying to prove in that research article. So when you understand the context of different authors, it becomes easy for you to discuss what the authors have actually discussed or written in those research articles. Now, by combining the author's viewpoint, you are discussing actually on a particular topic. You're presenting the facts side by side from different authors, and then you're providing your own viewpoint. So that's how a review article or a review paper is should be looked at. Now, coming to actual process of writing a review article. So before you actually start writing or before you actually start gathering the information, it's very important to know for which audience or which group of population or which group of authors you're actually writing your review article. So know your audience and their expectations. For example, let's say there is a particular journal, regulatory toxicology and pharmacology, and you want to write a review article for that particular journal. So go to the website of that journal, read what kind of content they're publishing. Okay, then try to see the different types of articles they're publishing. So if you see that, you will know then the boundary of the topics in which they publish first. Second is, what is their intent or object of that journal? So every journal does not publish, let's say, original research, or let's say on any field. So you have to know which particular audience or which particular group of population your review article is going to be published for. So that's the audience, their expectation, and that will set the perspective. Secondly, the purpose of writing. So for example, you want to publish a review article on, let's say, toxicity of vitamin D. Okay, so that might be the topic of your review article. Now, that's the objective, to discuss about the toxicity of vitamin D. Now, why are you writing that review article? Is there a need for writing such a review article, or is there recent changes in the research on vitamin D which triggered your writing? Or let's say, is there a recent discussions in the scientific community about whether vitamin D should be prescribed on a daily basis, or let's say on a weekly basis, or for a particular population in a particular country? So that will give you the purpose of writing. So you have to know your purpose. Second. Third is organizing your thoughts even before gathering the information. That is very essential, and you should actually spend a lot of time in gathering your thoughts even before searching for the information. Because if you do not gather your thoughts, or let's say if you do not have a clear outline in your mind what to gather or from which source or what type of information, you can spend n number of hours in reading or doing literature search gathering information from different websites. So it's very essential to have a clear outline in your mind. For example, recently I was working on a particular project. It's about trying to find out the toxicity of chromium oxide. Now, this chromium oxide is a pigment. It's a coloring pigment which is widely used in the industry, mainly in the cosmetic industry, but also as catalyst in chemical industry or in food. So it has widespread use across different industries and in different countries of the world. Now, the purpose why we started gathering information was to focus on a certain aspect of the toxicity of chromium oxide. So we have to gather our thoughts, how we will outline the review article or a review report. So it was not only gathering information on the toxicity of chromium oxide, let's say on reproductive toxicity, or let's say carcinogenicity or mutagenicity, but it was also to discuss what the regulatory authorities, for example, the Directorate of Drug Control of India, or let's say the US Food and Drug Administration, or let's say the European Commission. So these are the different regulatory bodies, what they have discussed on the toxicity of the chromium oxide. So the toxicity of chromium oxide was on one hand, then the discussion in the scientific community about chromium oxide toxicity is on the other hand, and then how the toxicity of chromium oxide could trigger a particular classification, or say particular classification of chromium oxide as a mutagen or as a carcinogen or as a reproductive toxicant. So the thoughts were actually on these few topics. So that was an example from one of my recent projects. So before, even if you do a literature search, it is very essential to have your thoughts clearly in your mind, do proper planning on what will be the aspect on which I will gather the information, right? Second is on which aspects I will focus my discussion. And then finally, when you have this clear outline of the article, and then you will clearly see that what would be the sources for your literature search. So there can be PubMed, or let's say Google Scholar or ScienceDirect. So n number of sources, but probably you're trying to find out the chemistry of a particular compound. So these particular journals may not help you gather those information. So you have to look for different sources of databases. So that's why it is very important to organize your thoughts, and then you will clearly see which are the databases you need and what type of information you need. So this is the organization part. And then before you start working, it's very important to to estimate or to imagine how much time you will need for a literature search, to gather the information, to review the information, to read through those articles, and then actually write. And then comes the review part, revision part, editing part. So you have to clearly divide those steps and estimate the time. This helps you proper planning for review article writing. So the planning is the most essential part of any kind of writing. Coming to the next slide. These are the few things which you need to remember when writing a review article. Obviously, the content of this short presentation has, what I have done is I have basically read different guidance, different guidelines, different discussions on how to write from different universities, institutes, and then gathered my thoughts. So these are not my own thoughts. I have only tried to provide a structure to the presentation, but these are all common thoughts of different authors. Okay, so starting with, as I mentioned initially, check the journal's aims and scope. So it's very important for you to decide the purpose of your writing and the audience, and then to decide upon which journal you are going to publish. Okay, so once you've decided the journal, you also need to check what are the aims and scope of the journal. I mean, what kind of information or what type of article they publish. So that's very important, because often what I've seen is articles written from a particular journal because they don't match the expectations of the journal, or they don't match the scope of the journal. So it's very important. Secondly, once you have seen the journal scope, you have to also define the scope of your article. Okay, so that will be quite important, knowing the boundaries of your own article. It cannot be, let's say, a 20-page or 30-page review article. It has to be limited in your scope. So you have to define the scope along with your teacher or your supervisor, your guide. Okay. Secondly, third point is finding sources of information. I mean, what will be the databases? For example, it could be PubMed, or it could be Science Direct, it could be Google Scholar. So finding out the right sources of information. Second is, next is spend time in writing an effective title, abstract, and keywords. They are the utmost important for getting your review article to the audience, because many journals are not open source journals, right? So when you publish your article in a journal, it might be paid article, and in most of the cases, the audience or the any student or teacher would only see the title, the abstract, and the keywords. So spend time in writing first the effective title. The title should convey the purpose of your writing as well as the scope of your writing and what you are trying to convey. So the title should be very effective, but not too long, not too short, but yet should give the meaning properly. Then the abstract. Most of the journals would have, let's say, a limit of, let's say, 500 words or 300 words for an abstract. So the abstract would basically come from your entire review article. What I have seen from many authors or many of my juniors is that they write the article and then they write the abstract. That should not be the way you do. You should first form your abstract, because the abstract directly comes from your thoughts. Okay, so when you have gathered your thoughts, you planned how the entire article should look like. You also know what would be the content of your abstract. And then the keywords. So the keywords should point to only the key message of your review article. So you have to also form the keywords very effectively and very planned way, so that you know that whenever you search for a particular review article or particular research article, you form your key strings, okay, in your, when you search. For example, let's say I'm going to search for acute toxicity and let's say calcium carbide. Now, if you do not have the keywords in the abstract, for example, acute toxicity or calcium carbide, but you have written something else, your article would never come in the search. So you will never get that article. So forming the keywords is very essential. Then introduce the topic and provide some context. Now, in the introduction part of a review article, you have to introduce the topic, that's understood, but you also have to provide the context. I mean, why you are actually trying to write on this topic, and what is the context, what triggered the writing of the review article. You cannot write for no reason, right? So there has to be some purpose. And also that there has to be some, let's say, that there is a recent discussions on glyphosate. Okay, I took this example from recent discussions in the scientific community. Glyphosate is a pesticide. It is used widely in the United States of America, in the European countries, and in the Asian countries by farmers across different countries and different areas. So it's a widely used pesticide. And glyphosate had very controversial toxicity coming from different studies. So glyphosate was widely discussed over the last 10 years, I would think. And recently, for the last couple of years, they are again being discussed whether to classify glyphosate as a carcinogen or not. Now, you could try to publish or write a review article on, let's say, glyphosate, but there has to be some context. So why you're writing? So the context would be the recent discussions on glyphosate and recent debates across different regulatory authorities on glyphosate use by the farmers. So that's the context. So you have to know the context and you have to introduce the concept and the context in the introduction part. Second, next critical thing is including a critical discussion in a review article. So, as I mentioned to you, you need to present the viewpoints of different authors of different research articles, but also you need to include a critical discussion about what the authors think, whether those viewpoints, which are those viewpoints are correct, and let's say how the viewpoints differ with each other. So this kind of critical discussion need to be a section in your review article. Finally, you have to summarize, let's say in a summary part or in a discussion part, and then even before you send it for review to the journal, there has to be rounds of review of your article, either by a friend who is very, very critical, or either by your teachers who are very critical, so who always criticizes work. So it's always good to know the viewpoint of your friends, your teachers, even before you submit the article to a journal for review. So these are the few things which you should always remember. Now, for the benefit of most of the students, I'm going to discuss about what are the common pitfalls. Pitfalls are usually errors, or let's say our prejudices on different topics or different sections of an article. For example, in the introduction, what we usually do is we write unnecessary background, or we repeat kind of sentences. This should be avoided, because you already did your basic research on the topic, you gathered your thoughts, so cut down the unnecessary background, cut down those repetitive words, be very specific what you want to discuss. So introduce the topic, but not by discussing unnecessary information. The information which should be discussed in the introduction has to be directly related to your topic. Second is exaggerating the importance of your work. So you can always emphasize or show the importance of why you are writing the review article, but do not exaggerate. And that could be done, let's say, at the end of the introduction paragraph. Not focusing on a clear and compelling research, question, or hypothesis. So a review article always discusses different authors or the viewpoints of different authors. So do not focus on a very particular research. So you have to open in your mind to discuss all the viewpoints. In the method section, what we usually tend to write always, and this method section is very relevant to a basic research article rather than a review article, is you reiterate meaning you rewrite the published method. It's no need to rewrite them. You can only cite them, or you can provide the name of the author. For example, such and such method or such and such determination was carried out by the method written by X et al. So you only cite the method, do not reiterate. Next big important thing is not to write a protocol, but to write a method. A protocol is a series of steps in a particular experiment. How do you do, and how in what order? So this is a protocol. When you write a research article or even do a review article, so in a review article, you can mention about the different methods adopted by or used by different authors. So try not to write a protocol, but to write a method. A method should be written as, let's say, in a past tense, and in general, what was the method followed. All right. So, and then check whether all the important methods or steps have been written in the methodology section. So for example, you're trying to find out the adverse effect or the toxicity of a particular compound on the fertility of, let's say, Drosophila. And when you write the method section, you have, let's say, did your research on different aspects. Let's say you have tried to find out the enzyme levels, you have tried to find out the cell viability, you have tried to find out the number of eggs a particular fly has hatched, you have tried to find out how many days the fly took to hatch the eggs compared to the control fly. So these are the different methods. Now, obviously, you cannot write the protocol. You have to write methods, highlight the methods. Let's say for enzyme, finding out the enzyme activity, finding out the cell viability. These are the methods used. So you know these are the methods. So 4 or 5 methods they have been used. Now, when you discuss the results in your results section, those methods and results need to correspond to each other. Always remember, if you have written a method for enzyme induction, you have to discuss the results of enzyme induction in the results section. So when you write the results section, always check what methods you have cited or what methods you have written in the method section. They should always correspond with the result section, and the result section would also look the same way as you have done your method. So following the order, that gives a good impression to the article that the sections actually correspond or match each other. All right. Then one more important point for the method section is to provide a context. For example, you're trying to, let's say you did an assay developed by so-and-so et al. What is that assay for? So the assay is for finding out the cell viability in the control flies and the treated flies. So you have to provide the context, not just the method. Otherwise, the reader is lost. Why you have done so many assays? So you have to provide the context for each of the method you write in the method section. The result section, again, coming to the result section, these are the some pointers. For example, overstating the results or reporting irrelevant results, or let's say not organizing them under different subheadings, or not including not including appropriate illustrations. So these are the few pointers. I'm sure you have written a lot of research articles. You know how they have been written, but try to remember these points. For example, when you do an illustration of a figure, check again and again whether the figure corresponds to the illustration. For example, the figure is talking about a graph of, let's say, number of cells counted over different time. For example, at 5 minute, 10 minute, 30 minute, 100 minute, so and so. So you have counted the number of cells over time. So the illustration in your figure should always correspond to what you are trying to show in the figure. So these are the few pointers for the different sections, remember. Coming to the figures, titles, and captions. For figures, often what we see is we provide a figure but do not mention that in the text. That should not be the case. Otherwise, the reader will be lost. What is that figure trying to convey? Then again, the data which you have written under the result section is not consistent or not matching with the figure. So let's say you are discussing about a decrease and increase, and again decrease about a particular value in a particular figure. you have to also discuss that in the text. All right. Then labeling is very important for a particular figure. So figure 1, 2, 3, 4, or ABC, however you want to label it, they have to be labeled properly and they have to correspond with the textual data. So these are the few pointers about the figures. And then captions. Normally, we do not write captions in very abbreviations or shorthand form. Never. Always try to write whole sentences, because if you write an abbreviation, make sure to include an glossary for abbreviations. That's very important. And many times I have seen writing, let's say, the concentration only in CONC dot. We all know what CONC dot means. It means concentration, but some of the readers might not know. For again, let's say you write LD50 or LC50. Most of the readers would know it's a lethal dose, median lethal dose 50, but some of the readers might not know. So you have to introduce a glossary section either at the beginning or at the end of the article to mention what those abbreviations are for. Remember, acronyms and abbreviations, they are two different things. Abbreviations are usually short form of an word. An acronym is usually a short form of a noun. For example, acronym of United States Food and Drug Administration is US FDA. That's not an abbreviation, that's an acronym. So clearly differentiate between these two in your articles. Coming to the discussion and conclusion section, including too much of information in the discussion does not help, or even in the conclusion. So whatever you have already discussed should be in the main body of the article. In the discussion, try to take only those arguments, or let's say only those points which are focused towards your topic, first of all, and second of all, which you have, let's say, decided at the end to conclude. So the conclusion or the discussion has to be focused. Second is failure to follow arguments which you have actually set up in the introduction. So the introduction and your conclusion should tally, or actually should follow each other. So what you have discussed in the introduction section also has to then follow a logical argument or logical flow in the conclusion section. Failure to focus on the current results. That's also one of the common pitfall of the conclusion. And improper tense. Normally, a conclusion is always written in a past tense, not in a present tense. And remember, one hedge word per sentence. Hedge words are usually like might be or maybe, could be. So in a sentence, when you are writing your conclusion, you are trying to, let's say, hypothesize something, that because of this, it might have been this. So in a sentence, do not write several hedge words. In the first section, you may have written maybe, and in the second section, again, could be. Don't write that. Journals do not like them. All right, so these are the few pointers. Remember, these are the errors which you should not do. Now coming to some tips. The first and foremost is clarity and not ambiguity. Always, when you write any section in your article, you have to choose words whose meaning are clear to you. You know the meaning. Otherwise, you don't write the word. Arrange always the words in a sentence very wisely or carefully so that it conveys the exact meaning. I have provided an example. Let's say in example 1, it is written in low water temperatures and high toxicity levels of oil. We tested how well the microorganisms survived. Now, this is one way of writing. The other way of writing is we tested how well the microorganisms survived in low water temperatures and high toxicity levels of oil. So it's a direct direct sentence. So there's no ambiguity. So always try to provide this kind of clarity in each sentence you write. Always try to be direct, all right? And don't twist a sentence too much. Otherwise, the meaning is lost in twisting a sentence. I have also seen often using pronouns like it, that, this, there. Don't use those kind of words. They are very ambiguous. For example, let's say you are discussing two different nouns in a particular sentence, right? For example, you are saying that US FDA and WHO has said this, comma, however, they have not finalized or concluded. So now the obvious question is, who is what? Who is they? So do not write these kind of pronouns in your sentences. When you have only one noun or one subject in a sentence, it's fine to use it or that or this. It's fine. Then we know what you're talking about. But when you have multiple subjects in your sentence, do not write. Also, do not also write repeatedly. For example, when you are discussing about a particular method, let's say you wrote about the advantages and disadvantages of the method, but do not always write this method, it provided, that provided. Try to write the method of this. So try to restrict the use of these kind of pronouns. Second point is avoid using of excess words, which makes it difficult. It doesn't convey the meaning properly. I will show you some examples in later slides. For example, here also I have written some examples for you. For example, made arrangements for. You can simply write arranged, and that would convey the entire meaning. Do not write excessive words. Secondly, made the decision. You can simply write decided. That conveys the entire meaning. So it helps you in two ways. One is limiting your word count. First. Second is you are actually very concise in your statement. So always remember, concise statements, concise paragraphs convey the meaning more accurately with more clarity rather than using too much of words. Again, you see another example, made the measurement of, or let's say perform the development of. Simply write measured, simply write developed. It carries the entire meaning. Do not use excessive words. Now some standard rules. So again, going back to the previous slide, you can find out n number of examples of these kind of pointers from different journals. For example, Taylor and Francis or Science Direct, or let's say even Intake Open. So all these journals, usually they have kind of guides for authors. And you can find these type of advices in guidances for authors, how to write. So you try to find out them from different journal websites, download them at one folder, compile them in a Word document for your own reference, and over time, over and over again, refer them so that you do not forget. All right. Now some standard rules. You all know that, for example, is ex gratia, e.g., right? Now there is a way you write. So it's not E comma G comma, or it's not E dot G dot comma. It's always E dot G dot. So there are some standard ways of writing. Then again, in a sentence, when you have multiple and, do not provide or do not put any comma before any and, because and is joining two different parts of a sentence, and there is no end. There is no pause. Then et al is to be written as either et al dot or et al dot comma. So both are allowed, and always italicize or making it italics, et al. Okay, but you have to put a dot at the end or dot and comma at the end, otherwise it doesn't convey the meaning. Always consult high impact factor journals. Your teachers and guides can always point out which are the high impact factor journals, and they have a list of abbreviations which are standard. So when you write for a particular journal, they might give you the list of standard abbreviations which they would like to follow. So when you write for a particular journal, follow those abbreviations. Some standard abbreviations, again, for some units which are followed worldwide, I have given here hour, minute, liter, or milliliter. These are very standard way of writing. So when you use them, try to use them in the standard way. All right. Now, again, for some other language tips, again, break out from the complex type of words, complex type of sentences. I have given some examples here. For example, like under the category, we'll see nouns or verbs or adjectives or adverbs. And commonly people, or I have seen authors writing, let's say, utilization or functionality, use simple words like use for utilization. It's the same meaning. Functionality, right? Feature. It's the same meaning, but a very simple word. Facilitate. You can write cause. Finalize. You can write end. Aforementioned, under adjectives, you can write mentioned, or you can write individualized. You can write individual. They are the same meaning. So why to use complex words? They will simplify your article, your discussion in a great way. And it will look very simple and convey simple meaning. So your article needs to be simple in writing or simple in reading, but it should convey the meaning properly or convey a good discussion. That's the point of writing an article, to write a critical discussion or to discuss the viewpoint. It's not to write heavy jargons or heavy words. No. Secondly, let's say there are some needless words. Often we write existing or we write already existing. So already you can delete. Existing means it is existing now. So already existing does not mean anything. Second, at the present time, not required. It's at present, always. Now, fundamentals. Basic fundamental doesn't mean anything. It's fundamental is always the basic thing. So basic fundamental, never use them together. Then eliminate. It's completely eliminate does not mean anything. Elimination is actually elimination, or completely eliminating from your body, or eliminating from your system. So eliminate is the word. It's not completely eliminate. Persist. So still persist? No. Persist is the present, which is actually present at a given point of time. So still persist does not mean anything. There are other words like mixed together, private industry, empty space. So all those red words are actual words which convey the meaning, and whatever you see in black, they're needless words. So mixed together does not mean anything. When you mix, you're actually mixing 3 or 4 things together. So together is an additional word, not required. So this is my last slide actually. So I can, let's say, keep on presenting these type of tips or these type of jargons, how to write clarity for hours and hours, but that was not the focus of this discussion. The focus of this discussion was to help you how to gather your thoughts, how to plan an article, what are the things to remember, and what is the standard way of approaching. So as I again advised you, go to the websites of those journals, for example, Taylor and Francis or Science Direct. Explore the websites. You will find guidances for writing review articles, guidances for writing a basic research article. Those are standard guidance, and you need to follow the guidance. And at the end, I have given here 4 references which I consulted. The Craft of Scientific Writing is actually a quite good article. Then the first one, which is written by Nicole Kelly from MIT. They have a program for writing and humanistic studies. This program is actually quite good. They have published a lot of articles on how to write, and you will find many of the slides or many of the information in this presentation from the Nicole Kelly presentation. Then there is a 1990 article, in 1990 published article from the American scientist, The Science of Scientific Writing. It's actually quite an old article, but it's helpful. And there are good guidance in the last reference. Sorry for the typo. You will see you'll see a dot after before the reference. It's Belcher et al. Is a course material for laboratory fundamentals. All these articles are freely downloadable, so you can download all these articles and you can consult them. So have them printed for your own reference, read them, understand, and finally try to imbibe in your writing. So that's my takeaway from here. All right, so I will now stop sharing. So I'm open for any question or discussion, although I'm not at all an expert in writing. I just learn each day and try to present you whatever I've learned and whatever I follow.
Thank you very much, sir. I would like to express my sincere gratitude to our resource person, Mr. Sobbosati Chakraborty, from our entire organizing committee of International Lecture Series on Nutrition for your presence and wonderful speech in lucid language. This is a very creative, well-organized, and student-oriented lecture. We are really honored and grateful to you for joining with us in our joyful learning program. We are running short of time, so let's move on to the next program. We omit, sir, our question and answer session. We will write to you in through mail if there is any question arise. So
Sure.
our next, thank you, sir.
Thank you, Saurav. So I'll be leaving because I have, as you know, it's a work day, so I have to leave for my work. So I'll be again happy to discuss with you or provide my guidance on any question your students might have. So again, thanks a lot, and we'll talk to you later.
Thank you, sir. Now, our last program is the vote of thanks for today by Sri Taraknath Dotto, Associate Professor, Department of Commerce and Coordinator of IQSC, Guskara Mahavidyalaya. Over to you, sir.
We have arrived at the concluding moment of today's program of International Lecture Series on Nutrition. Now we can confidently say that it has been a successful event. On behalf of Guskara Mahavidyalaya and Internal Quality Assurance Cell, Guskara Mahavidyalaya, I express my sincere gratitude to our learned speakers for kindly agreeing to take part in the lecture series. Dr. Abhijit Majumdar, Professor, Department of Zoology, Bardhaman University, and our keynote speaker, Dr. Sumedha Rai, formerly of the Department of Zoology, Bardhaman University, and at present research scientist, Ghent University, Belgium, Mr. Sabyasachi Chakraborty, senior consultant relating to human health both in India and abroad. Your deliberations have substantially enriched both our teachers and our beloved students, and also our understanding of some significant areas of knowledge. We are also thankful to other resource persons and teachers present here who will take part in this lecture series in coming days. I would like to thank our beloved principal, Dr. Swapan Kumar Pan, without whose active help and support this lecture series for the students would not have been possible. This online lecture series has been a collective endeavor in which students of many colleges and institutions, including ours, have participated. I thank them all. I would like to thank all members of the organizing committee of this series, including Dr. Saurabh Sarkar, convener, joint conveners Dr. Sukhendu Rai and Sri Sontu Nandi, JAL members Professor Rubinur Khatun, Mrs. Aparnita Nandi Rai, Dr. Nabendu Rokhit, and Ms. Paulmi Rai for their hard work to make this web lecture series a successful one. I express my sincere thanks to Mr. Brajendranath Adhikari, our office colleague and technical support staff of the program. Finally, let me mention that this lecture series is primarily intended for undergraduate students. It is my pleasure to thank all participants, including beloved students, both of Guskara Mahavidyalaya and of other colleges, and also all our teaching and non-teaching staff for their enthusiastic participation. Thank you all. সবাই সুস্থ থাকুন, ভালো থাকুন। Thank you.
এই সেশনটা শেষ করে দাও আর কি, এরপরে পরের সেশন হবে।
আমরা আজকের দিনের মতো এই সেশনটা আমরা এখানেই শেষ করছি। পরের সেশন গুলো আমরা যথারীতি যেরকম করে প্রত্যেকজন পার্টিসিপেন্টের কাছে লিংক আমরা প্রোভাইড করি, ঠিক সেভাবেই প্রোভাইড করব। আজকের মতো সেশন আমরা এখানেই শেষ করছি। ধন্যবাদ।
বলছি সুখেন্দু, বলছি আমাদের পাশেই তো মানপুর ওদিকে দেয়া যায় কোনোভাবে? মানে মান করে যেমন সুকান্ত বলছিল
হ্যাঁ
যে
স্যার।
স্যার আমাদেরকে যদি স্বপনদা পাঠানো হয় তাহলে আমরা দেখি কাউকে।
হ্যাঁ স্যার, অবশ্যই স্যার, আমাদের ওয়েবসাইটেও লিংক গুলো আছে, প্লাস আমরা আমরা দেখি, বাট আমরা পাঠানোর ব্যবস্থা করছি স্যার।
হ্যাঁ হ্যাঁ, আচ্ছা আচ্ছা, মানে আমাদের ওয়েবসাইটে আছে, তাহলে তো ওদিকে বলার কিছু থাকে না।
হ্যাঁ।
হ্যাঁ,
আমাদের ওয়েবসাইটে তার সাথে আমরা স্যার একটা মানকর কলেজে একবার ফ্লায়ারটা আমরা পাঠিয়ে দিতে পারি স্যার।
আচ্ছা আচ্ছা, তাহলে
স্যার,
মানকরকে একটা পাঠিয়ে দাও, উইমেন্সকে একটা পাঠিয়ে দাও। আর কাদের আছে নিউট্রিশন?
স্যার স্যার আমি একটা কথা বলি, আমি
হ্যাঁ।
প্রত্যেকটা কলেজকে নিজে তাদের যে হেড আছে নিউট্রিশন ডিপার্টমেন্টকে ফোন করেছিলাম, তার মধ্যে ওমেন্স কলেজ, হাটগোবিন্দপুর মেমারি কলেজ, ইটাচুনা কলেজ, হুগলি ওমেন্স, হরিপাল কলেজ এবং বেঙ্গাই কলেজে প্রত্যেককে ফোন করেছিলাম, কিন্তু মানকর কলেজের ওয়েবসাইটে কোনো ওদের নিউট্রিশনের কারোর নম্বর বা কারোর কোনো ডিপার্টমেন্টে কিছু পাইনি, তাই মানকরকে একমাত্র আমি জানাতে পারিনি। তাহলে আমি প্রত্যেকটা কলেজকে ইনফরমেশন দিয়েছি যাদের নিউট্রিশন আছে, এবং তাছাড়াও বেশ কিছু জনের জিওলজি বা অন্যান্য ডিপার্টমেন্ট যারা আছে, আমি তাদের ডিপার্টমেন্টের হেডদের নাম খুঁজে পার্সোনালি ফোন করেছি এবং স্টুডেন্টদের জয়েন হওয়ার জন্য আমি নিজে তাদের পাঠিয়েছি। যেমন মেমারি কলেজের ওয়েবসাইটে ছিল না, কিন্তু আমি পার্সোনাল কন্ট্যাক্ট থেকে তার নাম বার করে আমি ওই দেবলীনা আছে তাকে ফোন করে তার স্টুডেন্টদের জয়েন করেছি, কিন্তু মানকর কলেজের অ্যাক্সেস আমি করতে পারিনি স্যার।
ঠিক আছে, আমি মানকর কলেজের নাম্বারটা তোমাদের দিয়ে দেবো এখনই।
আচ্ছা
আর
স্যার।
আরেকটা হচ্ছে সেটা হচ্ছে যে যত নাম্বার অফ বেশি কলেজ থাকবে, বিশেষ করে আমাদের বাইরের স্টেটের যদি একটা আধটা কলেজকে যোগাযোগ করে নিতে পারো না, ছাত্র যেমন ধরো রিসোর্স পার্সন আসছে, তাহলে আরো ইয়েটা গড়ে যায়। এই তো আই.এম. নাসরিন আছে, ওই যে উইমেন্স কলেজের আছে দেখছি।
স্যার আমাদের কলেজে 33 জন স্যার, বাকি তো সব অন্য কলেজেরই। আমাদের কলেজে
আচ্ছা, আমাদের
33 জন
কলেজের
স্টুডেন্ট।
যারা স্টুডেন্ট তাদের অ্যাটেন্ডেন্সটা ফ্রম দি ভেরি বিগিনিং টু দা এন্ড মানে কম্পালসারি করবে, কারণ এই ধরনের সিরিজে এত ভালো ভালো লেকচার হচ্ছে, তারা অন্তত শিখুক দেখুক। এটা তো, আর তোমরা জুলজি ডিপার্টমেন্টের সমস্ত ছাত্রদের নিয়ে নাও, জুলজি ডিপার্টমেন্টের ছাত্ররা, বোটানির ছাত্ররা, কেমিস্ট্রির ছাত্ররা আসতে পারে। এ তো যেভাবে লেকচার হচ্ছে, যে কোনো স্টুডেন্টদেরই এটা বিরাট কাজে লাগবে, তাই না?
স্যার একটু আমাদের একটু অসুবিধা আছে, সেটা হচ্ছে আমরা 100 বেশি জয়েন করাতে পারবো না গুগল মিটে। এবার আজকে ইউটিউব লিংক ব্রজেনদা ক্রিয়েট করে দিয়েছেন, কিন্তু রোজ যেহেতু আমাদের বিকেলের দিকে বা সকালের দিকে, সেই কারণে এই ইউটিউব লিংক ক্রিয়েট করাটা একটু অসুবিধা। তার আমরা একটা অল্টারনেট বার করেছি, সেটা হচ্ছে আমরা আমাদের রেকর্ডেড ভিডিওটা আমাদের যে নিউট্রিশনের ইউটিউব চ্যানেল আছে সেইখানে আমরা সেশনের পর আমরা আপলোড করে দেবো, স্টুডেন্টরা দেখতে পাবে, কিন্তু সেখানে ইন্টারাকশন করা যাবে না। পরে তারা অন্তত লেকচারগুলো পাবে যারা আমাদের রেজিস্টার স্টুডেন্ট বা বাইরের স্টুডেন্ট এমনি।
না, আমাদের ইউটিউবের লিংকটা অসুবিধা কি হবে? সুকেন্দু পারবে না?
হ্যাঁ স্যার, হ্যাঁ, ইউটিউবটা আমি শিখে নিয়েছি,
ব্রজেনের কাছে শিখে নেবে তো, ব্রজেনের কাছে শিখে নিলেই সুকেন্দু পারবে। হ্যাঁ, কারণ তাহলে যদি এই ইউটিউব লিঙ্ক হয়, আর সত্যি কথা ব্রজেন তো পরশু থেকে ফর্ম ফিল আপও চলবে, তখন আরেকটু ব্যস্ত হয়ে যাবে। তাহলে সেক্ষেত্রে ব্রজেনের কাছ থেকে তুমি একটু শিখে নাও, মানে জেনে নাও, জেনে নিয়ে তারপরে এটাকে তো আমাদের নিজেদের কন্ট্রোলে নিতে হবে। আর মানকরের প্রিন্সিপালের নাম্বারটা আমি পাঠিয়ে দিচ্ছি, সৌরভ সুকেন্দু দুজনকে পাঠিয়ে দিচ্ছি।
ঠিক আছে
আমি এখন দেখে তোমাদের বলবো।
স্যার প্রিন্সিপাল স্যারকে ফোন করবো না নিউট্রিশনের কাউকে করবো?
না, প্রিন্সিপালকে ফোন করলেই, আমি প্রিন্সিপালকে ফোন করলেই প্রিন্সিপাল তো বলে দেবে, মানে ওরা আমাদের নিয়ারেস্ট নেইবার তো, ওদের
ঠিক আছে স্যার।
আচ্ছা,
সৌরভ নাসরিন ম্যাম কিছু বলবেন না তো?
হ্যাঁ নাসরিন, আমাদের যে সার্টিফিকেটের ব্যাপারটা আছে, সেই সার্টিফিকেটটা সবার যখন পুরো কোর্স কমপ্লিট হয়ে যাবে, তারপর আমাদের একটা ফিডব্যাক ফর্ম থাকবে এবং তার সঙ্গে এক্সামিনেশন হবে। ওই ফিডব্যাক ফর্মের মধ্যেও কিছু কোশ্চেন থাকবে, সেইটা ফিল আপ করে দিতে হবে এবং তার জন্য টাইম থাকবে। সেই দিন আমাদের একটা প্রিন্সিপাল স্যারের ইচ্ছা অনুসারে আমরা একটা ফিডব্যাক ফর্মে একটা এক্সামিনেশন পারপাস করেছি, এবং এক্সামিনেশনটা দেওয়ার পর তোমাদের ফিডব্যাক ফর্মের সঙ্গে যে তোমরা নাম রোল নাম্বার দিয়ে এন্ট্রি নেবে বা রেজিস্টার্ড করেছো, সেই অনুযায়ী তোমরা সার্টিফিকেটটা তারপর পাবে। অর্থাৎ যখন শেষ হয়ে যাবে আমাদের প্রোগ্রাম 14 তারিখের পর, তারপর থেকে 15 দিনের মধ্যে আমরা 2 উইকসের মধ্যে আমরা এটা পাঠাতে পারবো। যেহেতু এখন পরীক্ষার সিজন চলছে বা পরীক্ষা শুরু হয়ে গেছে আমাদের, হয়ে যাবে তারপর আমাদের 2 উইকস সময় লাগবে টোটাল যে সার্টিফিকেটটা পাঠাতে।
ঠিক আছে, আজকের মতো প্রোগ্রামটা এখানে আমরা শেষ করি। বাকি যার যা ইনফরমেশন, যার কিছু জানার আছে, আমাদেরকে আলাদা করে গ্রুপ রয়েছে, গ্রুপে জিজ্ঞাসা করে নিলেই হবে। ঠিক আছে, থ্যাংক ইউ অল, থ্যাংক ইউ ফর ইউর প্রেজেন্স।
আর একটা কথা বলে দিই একটু সুখেন্দু, সেটা হচ্ছে অনেকে তোমরা যেটা করেছো সেটা হচ্ছে গ্রুপে অ্যাড হয়েছো কিন্তু অনেকে ফর্ম সাবমিট করোনি। বন্ধুদেরও বলবে যদি ফর্ম তোমরা সাবমিট না করে থাকো তাহলে কিন্তু তোমরা রেজিস্টার হলে না আমাদের কাছে। যেমন আমাদের উদাহরণ হিসেবে বলি, আমাদের 240 থেকে 45 জন কিন্তু দুটো আমাদের ইউটিউব লিঙ্ক দেওয়া ছিল, তার মধ্যে দেখবে দুটো গ্রুপে তোমরা জয়েন হয়েছো, কিন্তু তোমাদের মধ্যে 171 জন মাত্র দেখা যাচ্ছে রেজিস্টার্ড হয়েছে, অর্থাৎ বাকিরা রেজিস্টার হওনি। তার কারণ হচ্ছে তোমরা যেই ইউটিউব লিঙ্কে জয়েন হয়েছো তারপর ইউটিউব জয়েন হওয়ার পর ব্যাক করে আর সাবমিট করোনি। এবার ব্যাক করে এসে সাবমিট না করলে কিন্তু আমাদের কাছে রেজিস্টার্ড হলে না, রেজিস্টার্ড না হলে কিন্তু সার্টিফিকেট পাওয়া যাবে না। তোমরা বুঝবে কি করে তোমরা রেজিস্টার হয়েছো কিনা, সেটা বুঝবে যখনই তোমরা সাবমিট করবে এবং রেজিস্টার্ড ক্যান্ডিডেট হবে, তখন সেই একটা ফর্ম তুমি যেটা ফিল আপ করেছো তোমার কাছে কিন্তু, তুমি যে ইমেল আইডি দিয়ে ফর্ম ফিল আপ করেছো তোমাদের কাছে কিন্তু চলে যাবে। অর্থাৎ তোমার কাছে যদি ফর্ম ফিল আপ করা একটা কপি আসে যে তুমি কি ফর্ম ফিল আপ করেছো, তবে সেটা তুমি কিন্তু রেজিস্টার্ড ক্যান্ডিডেট হলে। যদি সেটা না থাকে তার মানে তুমি জানবে তুমি শুধু ইউটিউব লিঙ্ক অব্দি অ্যাড হয়েছো, তার বেশি কিন্তু হওনি, তোমরা কিন্তু রেজিস্টার্ড হওনি। তোমরা অ্যাকর্ডিংলি সেই অনুযায়ী কিন্তু স্টেপ নেবে। যদি না হয়ে থাকো তাহলে কিন্তু রেজিস্ট্রেশনটা করে নেবে, আদারওয়াইজ কিন্তু আমরা সার্টিফিকেট জেনারেট করতে পারবো না, কারণ এটা হচ্ছে অটো জেনারেট, অর্থাৎ নিজে নিজে জেনারেট হয়ে যাবে। তাহলে আজকে আমাদের সেশন এখানে আমরা শেষ করছি। তোমাদের নেক্সট সেশন তোমাদের গ্রুপে জানিয়ে দেওয়া হবে নেক্সট ডে গুলো। হ্যাঁ, সুখেন্দু অফ করে দাও।
Okay.